All right. Hi, everyone, and welcome to today's webinar, Advancing Cancer Organoid Drug Screening with Novel Media Systems. I'm Colin Paul. I'm a staff scientist in cell biology R&D at Thermo Fisher Scientific. Today's event is presented by LabRoots and brought to you by Thermo Fisher Scientific. Our webinar today is a two-part presentation. I'll be starting us off with a look at how tumoroids can be readily cultured and scaled using Gibco OncoPro tumoroid culture medium. Dr. Christophe Deben from the University of Antwerp and Orbits Oncology will then share his work on methods to advance tumor organoid drug screening. For complete biographies of our speakers, please visit the Biography tab at the top of your screen. We encourage you to participate today by submitting any questions you may have during the presentation. To do so, simply type them into the Ask a Question box and click Send. We will answer as many questions as we have time for at the end of the presentation. Also, if you experience any technical questions, you can utilize the Q&A box and we will assist you directly. So we're glad you're all here. I will start us off with the first presentation on enabling tumoroid screens using OncoPro tumoroid culture medium. I'm excited to present how Gibco OncoPro tumoroid culture medium can enable tumoroid-based compound screens. The OncoPro medium kit, shown here, is designed for the culture of patient-derived tumoroids from epithelial cancer indications, including colorectal, lung, head and neck, pancreas, endometrial, and breast cancers, and makes physiologically relevant patient-derived tumoroid culture more accessible and standardized. Our interest in patient-derived cancer models has been driven by the recognition that while cancer is responsible for around 10 million deaths worldwide each year, development of an anti-cancer therapy typically requires around 10 years and $1 billion US. Some of these costs occur because the majority of compounds that look promising in preclinical research fail during clinical trials. Today, we'll focus on the use of physiologically relevant patient-derived tumoroid models in the development of anti-cancer compounds, which could include small molecule inhibitors, cytotoxic drugs, targeted protein degraders, biologics, antibody-drug conjugates, and hormone therapies. As these compounds are evaluated functionally for their effects on cancer cells, the development of accurate and representative in vitro cancer cell models is crucial for understanding tumor biology and improving therapeutic strategies. A number of the approaches discussed here can also be used in the evaluation of potential cell therapies with these cancer models. Traditional immortalized cell lines have limitations in mimicking the complexity of patient tumors, including properly representing gene expression patterns and clinically observed mutations. Primary patient tissue and animal models provide greater biological complexity but can be difficult and expensive to source and maintain. In contrast, patient-derived tumoroids or cancer organoids are obtained directly from patient samples and grown in 3D culture systems using specialized serum-free media such as OncoPro tumoroid culture medium. Compared to immortalized cell lines, tumoroids more accurately reflect the biology and heterogeneity of patient cancers, have scalable growth, and importantly retain the genetic and phenotypic characteristics of the original tumor to enable use throughout the drug discovery pipeline. Today, I'll discuss 5 steps to enable tumoroid-based compound screens, including sourcing tumoroid models, culturing and characterizing tumoroid cells, optimizing conditions for compound screens, and running assays using different analysis modalities. Let's step through each of these in more detail. Step 1 is to source tumoroid cells for use in compound screening assays. I'll focus on examples of colorectal cancer, or CRC tumoroid lines that were derived from human cancer cells using OncoPro tumoroid culture medium. Our typical workflow involves dissociating fresh resection samples, aggregating cells in suspension for 2 to 7 days, embedding tumoroids in basement membrane extract or BME domes for initial growth, and then expanding tumoroids in suspension culture for bank generation. Additional reagents for handling tissue and deriving tumoroid lines are listed here, and Thermo Fisher has an application note available that describes this process in more detail. While new tumoroid lines can be derived from tissue samples, we also offer a number of Gibco OncoPro tumoroid lines across cancer indications that have documented 3D morphology, genetic and phenotypic stability, and consistent growth profiles, and can be used to quickly implement physiologically relevant tumoroids in assays of interest. During tumoroid line derivation, a common challenge using culture media systems designed for adult stem cell organoid culture is the expansion of non-cancer cells. These healthy cells can sometimes overtake cancer cells in culture. OncoPro tumoroid culture medium was designed for the expansion of epithelial cancer cells and designed to not promote healthy cell growth. And we've seen from flow cytometry and RNA sequencing data that cancer cells are selectively supported over time. Stromal and immune cells, for example, that are present in the initial tumor samples eventually die off, as you can see by decreases in CD45 and CD31 protein levels by flow cytometry between the initial tumors and established tumoroids. Importantly, tumoroids derived in OncoPro tumoroid culture medium retain donor-specific mutational profiles and gene expression patterns. Targeted sequencing using the OncoMine Comprehensive Assay V3 shows that both the presence and frequency of single nucleotide variants and key oncogenic mutations are retained when comparing initial tumor samples to derived tumoroids across a range of genetic backgrounds and different mutations. The plot on the left shows matched single nucleotide base substitutions and oncogenic mutations between colorectal tumor samples and derived tumoroids, where mutations are consistently conserved in culture. Additionally, RNA sequencing reveals a high concordance of gene expression levels between initial samples and tumoroids across indications. The heat map on the right shows a comparison of tumor samples to tumoroids for select colorectal, lung, and endometrial cancer donors. Overall, we see a typical correlation in gene expression patterns between starting material and tumoroid cells of around 85%, and most of the difference is due to the loss of immune and stromal cells over time. After tumoroid cells are growing stably in culture, they can be expanded and banked. OncoPro tumoroid medium was developed for compatibility with a suspension culture approach, where tumoroids are grown in 3D in the presence of a small amount of basement membrane extract protein, but handle much like a suspension cell line. We do this while retaining the morphology and gene expression patterns that are observed in embedded culture within hydrogel domes. And a lot of our culture involves some of the reagents listed on the bottom right of this slide. This enables generation of large banks of tumoroid lines, which we typically feed every 2 to 3 days and passage every 4 to 14 days. And we call these lines established once they have these stable growth rates, demonstrate at least 5 cumulative population doublings from the original cell number, and are bankable and freeze-thaw competent. Importantly, the morphology of these tumoroid lines are donor-specific, as you can see on the images on the left, and maintained over passages in vitro. These also grow at stable rates both before and after cryopreservation, which is indicated in the plot in the middle of this slide by arrows for freeze-thaw cycles. And in many cases, this growth is stable over 250 days. These lines also exhibit distinct subpopulations within the tumoroids as measured by single-cell RNA sequencing to show evidence of tumor heterogeneity. After scale-up, we freeze tumoroids in recovery cell culture freezing medium to establish working banks that can be pulled from for downstream assays. Characterization is very important for these patient-derived tumoroid models. The key draw of these models is that they look like patient tumors. We frequently perform next-generation sequencing to compare early tumoroid banks to tumoroids that have been cultured in vitro for longer periods of time. In many cases, this involves use of the UAntwerp OncoMine Comprehensive Assay for mutational profiling and the Ion AmpliSeq Transcriptome Human Gene Expression Panel to analyze gene expression patterns, as well as routine monitoring by microscopy and at times flow cytometry. This type of analysis has demonstrated high concordance between early banks of tumoroid lines and cells in culture, in many cases for up to 250 additional days. Correlation analysis of the frequency of single nucleotide variants, or SNVs, identified using the OncoMine panel show high correlation, these are all at 0.98 and above, between tumoroid banks at passage 13 and tumoroids after 53 to 258 additional days in culture. Additionally, this is a nice fingerprinting approach, as you can see that there's very little cross-correlation of SNV patterns between donors. Consensus molecular subtyping from gene expression data additionally demonstrates that colorectal cancer subtypes are maintained in a donor-specific manner long-term. You can see 3 donors on this slide. That occurs at different passage numbers as shown here. And a diversity of these different molecular subtypes are supported by the media system. Principal component analysis and differential gene expression analysis tell a similar story. Global gene expression patterns are conserved during long-term culture, with tumoroids at multiple passages in the PCA plot on the left clustering by donor and not converging on any common phenotype. Additionally, differential gene expression analysis between cultures at passage 13 and at passage 50 show significant changes in less than 3% of over 17,000 genes, and these changes are associated with minimal significant changes in KEGG pathways. Overall, we've seen that tumoroid lines established using OncoPro tumoroid culture medium are quite stable in culture. Optimization of tumoroid cultures for downstream assays involve both scale up to generate cell numbers required for different assays and scale down, typically to 96 or 384 well plates to achieve the required throughput. We typically use non-tissue culture treated flasks for scale up. These flasks are well suited to suspension culture with OncoPro medium and are available in a number of sizes. In particular, the suspension culture approach in standard flasks enables researchers to generate the cell numbers they need when they need them. Tumoroid morphology and growth rates, as population doublings per week here, are consistent from T25s up to T175s and triple flasks, and millions to tens of millions of cells per flask can be harvested to dissociate and seed into well plates for screening and analysis. This lets researchers maintain the biological relevance of tumoroid cultures while benefiting from the ease of use of the OncoPro medium system and the suspension culture approach. We routinely employ suspension culture to generate cells for downstream assays instead of having to individually plate tens to hundreds of basement membrane extract domes during scale up. After scaling up, we'll typically dissociate tumoroids and seed cells into 96 or 384 well plates for high throughput testing. Measurements of cell viability after plating show low variance across plates for multiple tumoroid lines and indications, and this variance can be further minimized by incorporating automated fluid handling during cell seeding, as you can see in the plot on the left. Overall, our guidance is to anticipate needing around 1.5 million cells per plate, with 12 to 15,000 cells per well in a 96 well plate and 3 to 5,000 cells per well in a 384 well plate. Formats such as microcavity plates, which include micropattern divots at the base of each well, can be used to further control tumoroid size. As far as best practices, we typically will strain cells through a 100 micron mesh cell strainer prior to plating and after dissociation to remove any non-dissociated cell clusters and promote even well-to-well seeding. One other watch out is to minimize cell settling during handling by using agitation or trituration, as these tumoroid cells tend to settle out of suspension more quickly than immortalized cell lines. Our typical timing involves seeding dissociated cells, culturing for 3 days to form tumoroids, and then incubating tumoroids with compounds from day 3 to day 7. Images of tumoroids at day 3, the time of compound addition, are shown here. At this time point, we observe low well-to-well variability across a given plate after following the cell seeding best practices discussed on the previous slide. Step 5, now that you have cells in a plate, is to run assays to detect and measure cell viability in response to compounds. I'll show examples of plate reader-based screens, which provide rapid readouts on a per-well basis, as well as high-content image-based screens to gain more information on cultures and improve amenability to space and time-resolved analysis. Thermo Fisher Scientific offers a number of instruments and reagents to facilitate cell analysis, including the various ScanLux multi-mode plate reader and the Cell Insight CX7 laser high-content analysis platform. One reagent that has worked particularly well for us in 3D cultures is called out on this slide, the Cell Event caspase 3.7 detection reagents, in which a fluorescent signal is activated by caspase 3.7 activity during apoptosis and is compatible with both live and fixed cells. As an example, one plate layout we've used for a compound screen in a 96 well format is shown here, though other configurations are certainly possible and may be better depending on the experimental situation. Our typical layouts involve serial dilutions of compounds with analysis across multiple tumoroid lines, with 2 to 3 wells per condition, as well as positive and negative controls. I'll show a pretty simple example using staurosporine in both plate reader-based and image-based assays in the next slides. For plate reader-based assays, we prefer to multiplex orthogonal measurements of cell viability to assess dose response to compounds by a few different methods. Assays can be run in about a week, as shown in the flowchart at the top of this slide. One analysis protocol involves incubation with Presto Blue HS or high sensitivity viability reagent, which is most accurate for 3D models if it's incubated overnight. You can then read that plate and then sample the supernatant to quantify lactate dehydrogenase release from dying cells via the Cyquant LDH cytotoxicity assay. And finally, lyse the cells remaining in the plate to quantify ATP levels at the different treatment conditions. Some example dose response curves to staurosporine using patient-derived colorectal tumoroid models as well as HCT 116 immortalized colorectal cancer cells are shown on this slide. One thing that's been interesting is that we see a highly heterogeneous response with these physiologically relevant tumoroid models. The HUCO 031721 model, shown in orange, for instance, shows marked resistance to staurosporine treatment, while the other tumoroid models generally are more sensitive than HCT 116 cells. In general, dose response trends are comparable across assay readouts, though IC50 values can vary depending on the mechanism of action of the detection reagent. I won't spend much time here, but one nice thing about the OncoPro medium system and its built-in suspension culture approach is that it's quite amenable to automated fluid handling. We can run these assays using an automated liquid handler and achieve comparable response as when we run the assay manually, but we gain the benefits of having reduced hands-on time with automation approaches. We typically perform similar multiplexing for image-based fluorescence detection of cell viability. Staining with cell-permeable nuclear dyes identifies all cells, while combining both cell event caspase 37 green with Imagit TMRM reagent distinguishes apoptotic and alive metabolically active cells, respectively. Other dyes can also be added depending on the instrument setup. We found that these live cell compatible dyes give the most consistent results when they're co-incubated with cells and tumoroids for multiple days prior to refreshing the stains and imaging around day 7. This type of approach, when combined with high content analysis on platforms, such as the Cell Insights CX7, shows really clear distinction between live and dead cells when the dose of the compound is changed. So you can see these multicellular tumoroids changing from red to green on the images here as staurosporine concentration increases and apoptosis occurs. Additionally, imaging reveals heterogeneity in drug response, with both live and dead cells observed at intermediate drug concentrations. These dyes can also be read on plate readers equipped for fluorescence detection, but you can see from these viability curves that markedly better signal-to-noise ratios and this really nice crossover between live and dead cell populations are observed when image-based approaches are used. So for some of these in-depth analysis routines and experiments where you really want to know what's going on with the state of different tumoroids within a given well, we really like these image-based approaches. In summary, we're excited about the continued growth of tumoroids as relevant target populations in functional assays during drug development. And we feel that OncoPro tumoroid culture medium, some of the lines that we've established, and some of the protocols that I've pointed out here will make these types of experiments more accessible to the research community. So let's conclude. Tumoroids can serve as a more functionally relevant target in functional compound screening assays by representing a diversity of donor genetic backgrounds, having compatibility with automation, and then using multiplexed assay readouts to gain confidence in the results. A lot of the information I presented here is presented in more detail in an application note that's also available. Should mention that custom services, as well as the OncoPro tumoroid lines that we've established, can accelerate the use of tumoroids in different applications, and we'd be happy to discuss potential projects there. Going forward, we anticipate that tumoroid models will be used more and more in high-throughput compound screens, as well as functional genomic screening. And these types of models have already shown great promise in functional screening assays, even to guide cancer treatment selection. We're really interested to see how these may move more towards the clinic in the future. Overall, Thermo Fisher is excited to support your tumoroid work throughout the workflow, really from sourcing, culturing, characterizing, engineering, and assaying these tumoroid models. I'll wrap there, and thanks very much for your attention.
Hello everybody, thank you for joining this webinar, and today we'll talk to you about advancing tumor organoid drug screenings. My name is Christophe Deben and I work at the Center for Oncological Research at the University of Antwerp in Belgium. And as you can see, we're very closely located to the Antwerp University Hospital. This way we have easy access to fresh patient tumor tissue, allowing us to establish the UAntwerp tumor organoid biobank. And besides this biobank, we also now launched a new screening platform, DrugVision.AI, offering both confocal and wide-field imaging, all automated for advancing organoid drug screenings. So we're now capable of generating a lot of data, a lot of images. So the next bottleneck was image and data analysis. For this, we developed Orbits, an automated image and data analysis platform that we're now also making available to other researchers. So I want to start with a short introduction on patient-derived organoids. So the benefit is that we now can create these 3D tumor avatars from patients in the lab. And it has already been extensively shown that they can maintain a mutational landscape and clonal heterogeneity of the tumor found in the patient. And there's lots of applications possible. Preclinical applications, thinking of drug development, which has already been drastically accelerated using organoids, but also translational applications where we can grow organoids from a patient and predict therapy response. And also there's a lot of promising data out there now. The last benefit is that we can expand them, biobank them, share them, and really build extensive large-size tumor organoid biobanks in labs around the world. There's different types of tumor organoids. So the tumor organoids, or the tumoroids by themselves, are epithelial cancer cells. There's no tumor microenvironment, and they allow for long-term culturing and biobanking. Reconstituted organoids, or assembloids, allowing us to add fibroblast PBMCs and recreate the tumor microenvironment. And it's very interesting to see, for example, in these images, that the tumor organoids in red and the fibroblasts in green actually self-assemble into a microtumor over a course of a couple of days, which really resembles the morphology found in the patient tissue. And now with confocal imaging, we can also have an in-depth look at single cell level in these microtumor models. Lastly, there's patient-derived tumor fragments. So they really maintain a native tumor microenvironment, but the downside is that these are mainly short-term cultures and they cannot really be expanded that much. So it's more one single experiment assay, but there's a lot of different applications or protocols out there, for example, air-liquid interface, and you can even cryopreserve the tumor fragments and use them in later experiments. And this is especially useful in studying immunotherapy. Today, the focus of my talk will be mainly on the tumoroids or tumor organoids. So what are the challenges? So it's still quite challenging to establish and grow these organoids. It's very patient-specific, tumor type dependent, and short-term cultures often work better than long-term cultures, because often we see that organoids stop growing after 3-4 passages. It's also quite technical, it's expensive, it's complex media that you need, a lot of supplements, and you need quite specialized equipment and expertise to use these organoids. So this is also one of the reasons that often rudimentary viability assays are used, because there's not really the knowledge to do proper drug screenings. And for these two technical challenges, we actually aimed to create a solution. So when thinking of high-throughput organoid drug screenings, of course, ATP-based assays, even for low-throughput screenings, are mostly used, as you can see in literature. And the concept is that you treat the organoids, incubate for a couple of days, lyse the cells, measure ATP, and quantify relative viability. For us, this was not really useful because we won't know what's happening with the organoids during the treatment, and mainly because you don't know what type of effect your therapy is having. Is it growth arrest? Is it cell death? Is it a homo or heterogeneous response? And a lot of anti-cancer therapies actually affect ATP levels in the cells. So we really spent quite some time looking at images, improving the algorithm to detect the organoids label-free, and we now have a very robust algorithm that can detect different morphologies of organoids in different states of growth, therapy response, and so on. So now we have images, we have the raw data, but we need meaningful information, biologically meaningful information from these raw data. So the next step was to look into drug response metrics, and we used growth rate based metrics, and we saw that the ones that are used in literature are not really very accurate in showing what we see on the images. So we developed our own growth rate based metric, the normalized organoid growth rate. And for these growth rate based metrics, you can have a value between 1 and 0, indicating a cytostatic response, so growth arrest, or a value between 0 and -1, indicating a cytotoxic effect or cell death. So big benefit from one curve, one value, we can distinguish cytostatic and cytotoxic responses, which is of course crucial when looking at a therapy response and is completely missed when using relativity or, for example, ATP-based assays. So this is an example of organoids that are untreated. Organoids with an NOGR value of around 0, you can see mainly growth arrest. And when you have a negative value, there's a lot more cell death, still some viable organoids, and of course a value of -1 indicates that there's full organoid cell death. And the validation and the quantification of this metric is written in a paper now in a manuscript and will hopefully be available soon. So to give you a short overview of our drug screening pipeline, we culture the organoids in domes, so the regular method of culturing organoids, for 3-4 days. We harvest them, seed them with a pipetting robot, treat the plate with a digital drug dispenser. Then we of course use live cell imaging to capture the images, whole well images, and then use Orbits for the downstream image and also data analysis. So when we think of applications of organoids, and especially the ones we're applying in our lab, there's two main categories in my opinion. The first one being functional precision medicine. So the idea of growing organoids from a patient of the hospital in the lab, testing several therapies and giving the best performing therapy back to the patient. So this is also something we're retrospectively working on. And it's very important here to really build generate a lot of data so that you have organoids to compare, because screening one organoid doesn't really allow you to make classification if it will be a responder or not. So for pancreatic cancer, we're doing a retrospective study for each new successful pancreatic cancer organoid, treating them with gemcitabine or FOLFIRINOX. And then we can make a classification of resistant organoids, for example here, the intermediate responders, and the sensitive ones. And when we translate this back to the clinic, we can indeed see the ones that we classify as sensitive, here based on a derivative of the area of the curve of the NOGR of drug response curve. We can see the best responding one was indeed classified sensitive. An intermediate responder of 12 months progression-free survival was also classified as intermediate. And the worst performing patient with a progression-free survival of 4 months only, unfortunately, was classified as resistant. And this metric also takes into account the amount of cell death, which is completely missed with cell titer low. So we're still gathering the clinical data of some of these organoids. Some patients are still under treatment. So it's really a continuous process. You can expand your models and make them better over time. But what was very interesting to see actually is that in one well, from one patient treated one concentration of a drug, we see a very heterogeneous response. So there's resistant organoids, organoids with an intermediate response, so mainly growth arrest, and organoids that fully die. And actually, this ratio of sensitive versus resistant organoids correlates very well to the progression-free survival of these patients, and this is also validated and published. And again, it's quite a small cohort, and we're continuously expanding this. What I also think is a very promising field using organoids is making drug response prediction models. So we're clearly living in the age of automation, big data, and AI, and we can relatively affordable make molecular twins of patients' tissue, but also from tumoroids, for example, where we can collect the genetics or the genome, the transcriptome, the epigenome, kinome, anything is possible. And now in our lab, we also have the capacity to perform high-throughput drug screenings for hundreds of compounds on dozens of organoids. So we can generate a lot of data, drug response data, and link that to the molecular data of the patients to develop machine learning and deep learning models. So we can make molecular drug response models. For example, when we have the transcriptome of an organoid of a patient, we can predict using our models which therapy will work in these patients. Another strategy is to build these models using the molecular structure of a compound. For example, QSAR modeling, where you link the structure of a molecule to the activity, and without screening them, you can be able to predict the efficacy of a compound based on its molecular structure. So that's again also a very promising field, but needing a lot of data. So what are the challenges related to both of the applications? So functional precision medicine. For us, it's still unreliable success rates. So we never know from advanced if the organoids will grow from a patient, how fast they will grow, and so on. And we can only test a few therapies in a short time frame because, of course, if you want to test more therapies, you need more organoids, more time to expand them. It's also a very big issue is we can test anything in the lab, but that doesn't mean we can actually give these therapies to the patient because of reimbursement, insurance, and so on. And there's a big lack or large lack of standardization in culturing and also screening methods and analysis. For these DRP models, they're very data needy. And they also come with a high cost for developing these models, but still sequencing cost is still going down, and it will be more and more affordable to build these models, and they're much more robust. So I think a combination of functional precision medicine and DRP models can be very interesting, where we first identify effective drugs based on these DRP models, based on the molecular signature of the patient, for example, the transcriptome. Then the in silico hits we can test on the organoids, and then the best performing compound we can give to the patients. And eventually, once these models would be validated, the use or the need of organoids could be excluded, but we can still make accurate predictions of therapy response for these patients. So this is ongoing. I cannot show you any data for it yet, unfortunately, but I think this is can be a very promising strategy in the future. So in the second part of my webinar or my presentation, I want to talk about two solutions that tackle actually the lack of standardization in culturing and also the lack of standardization in drug screening, assays, and analysis. The first one is the OncoPro medium. So it's a new medium from Thermo Fisher that's developed to culture tumoroids, or the cancer organoids. And when we compare tumoroid medium, you of course have the conventional method or the hub method, where you have just medium, advanced TMM most of the time, where you add Wnt or spondin, noggin, a lot of growth factors, small molecules, B27, and so on. So it's very tumor type dependent, and also maintaining the stock of all these reagents is quite challenging, and often conditioned medium is used for spondin and noggin, which is also not a very standardized method. You can use recombinant proteins, but these are quite expensive. Then OncoPro medium, it's the basal medium, one supplement, BSA, B27, and depending on the tumor, you add 1 or 2 growth factors. So much less supplements, more standardized, and they're really trying to validate their medium on a lot of different tumor types. And also in our lab, we're testing several tumor types with quite some good success. So how do we do this in the lab? You have the conventional grown organoids. We transition them to the OncoPro medium. Slow transition, so 25, 50, 75, 100%. And then once they're fully transitioned, we collect FFPE samples, we biobank them, we collect RNA, DNA, and so on, and we also perform a drug screening. So it's ongoing. We cannot show any data yet on, for example, differential expression between HUB and OncoPro medium, which would be very interesting to see. But we do see quite some differences. So in the conventional medium, we see more rounded, smooth organoids, and in the OncoPro medium, they're much more rough. So there's a clear difference in morphology, which is also retained in a lot of other tumor types. When looking at the growth rates, so the images I showed you was from a cholangiocarcinoma. In growth rates, these organoids grew better in the OncoPro medium, but for other organoids, the growth rate can be similar. So it's again a bit tumor type dependent or patient dependent. But the most interesting part, so our expertise is really high-throughput drug screening, is looking at the drug response. There's quite a drastic difference between the drug response between the two media. And first I'm going to zoom in on the targeted therapies, or the TKIs, where on the left in the medium you can see there's a response to most of the TKIs, while on the right there's one clear hit being neratinib, so it's a pan-HER inhibitor. And when we zoom in on neratinib, you can clearly see that the OncoPro medium cultured organoids are more sensitive. You can see this on the dose response curve with the NOGR values, but also clearly on the images in the medium, there's quite a little response, while there's a lot of effects in the OncoPro medium. And also please note that the error bars in from our analysis, so we have high reproducible analysis in 3 biological replicates. When looking at other therapies, for example, chemotherapy, you can see that for 5-FU, cisplatin, the drug response is quite similar, but for other therapies, there's some variations. For example, in irinotecan, the OncoPro organoids are least sensitive, and HUB organoids are most sensitive, but for oxaliplatin, it's the other way around, where the OncoPro organoids are the most sensitive ones. For oxaliplatin, this is already known in the literature because NAC apparently seems to really induce resistance to oxaliplatin, like an antioxidant. So excluding this could maybe already sensitize the organoids in the hybrid medium. So what can we conclude from our couple of months working with the OncoPro medium? It has a lot of benefits because it's allowed for more standardization, it's ease of use, you need less supplements, your stock management is much more easy, and it's also actually much cheaper compared to the conventional method. But there's a lot of, on one hand, some similarities, but also a lot of differences with the conventional methods. So the success rate is also different. We're now doing fresh tumor samples in parallel. Some work better in OncoPro medium, other ones work better in the HUB medium. So as I showed you, clear difference in morphology, clear difference in drug response, and it will be very interesting to see which drug response actually correlates best to the clinical response in the patient. Not only interesting, but really crucial to see which medium that should be used. And I think, as I already showed, the difference is really therapy specific. So we need a lot of data comparing these two models. Then the second step for standardization is Orbits. So all the analysis that I showed you here, all the plots and so on, we really spend a long time developing, first of all, the image analysis part, so the label-free organoid segmentation that you can just use any wide field microscope, take a bright field image and segment the organoids. And the second part also fully automating the downstream data analysis and really focusing on clinically relevant drug response metrics. So the first part, image analysis, we also offer up to 80% data compression, so really saving a lot of storage, which is also a big issue when using live cell imaging. And now we're also developing a cloud-based data management and analysis tool, which I will show a little demo from afterwards. As I said, we offer pre-trained label-free tumoroid segmentation, and we are completely platform agnostic. So segmentation of organoids on an Inky site, Deacon Spark Cyto, a cell discoverer, these are the ones we have, but we can also apply these to other microscopes. We're also looking into reconstructing the organoids in 3D from, for example, Z-stack using bright field images, where you can really have the 3D morphology of the organoids. The algorithm also works well in microwells, ignoring the backgrounds, the form of the wells, but really only segmenting the organoids. And of course, we can also do fluorescence and tracking of the cells. Here we can see the formation of these microtumor assembloids that I previously discussed. So these are some of the things we're working on, but of course, we can do much more. If there's something specific you would need, you can still contact us. For the data analysis, thinking of drug screenings, quality control is of course important. So we automatically see or visualize the seeding density to see if it's consistent between all the columns and the wells or in the rows. We also quantify the Z factor. So this is quite standard metric that is used in drug screening, taking into account the dynamic range and deviation of the positive and the negative control. So a value closer to 1 is a very good screening assay. And we also detect outliers automatically based on the entire drug response curve rather than on, in this case, the 3 biological replicates, giving us a very robust and a robust method to exclude outliers without any user's bias. Like I already showed you several times in the presentation, we also developed a normalized organoid growth rate. We can calculate all its derivatives, the area of the curve, the energy R50, energy R max, and so on. This is all automatically calculated. We can do response classifications. In this case, we used the 30 percentile method to have 3 distinct classes of response. And we can also quantify synergy. So we included in the analysis automated calculation of HSA, Lua, Bliss, and ZIP synergy models. And we also spent quite some time in new ways to visualize synergy. And this is an example of a combination of a drug aranofin with an ACT inhibitor MK2206, where the bubble size indicates the amount of synergism. So a large size bubble means that there's a strong synergistic effect. So 1 + 1 is 3, 4, 5. And combining this with our NOGR metric, you can actually distinguish both cytostatic induction of synergy and cytotoxic induction of synergy. So a blue large size bubble would mean there's strong synergism, but there's not really a lot of happening. So maybe 5% + 5% is 15% of inhibition of cell growth, which is not really biological meaningful. So we would really be looking for a large size bubble, strong synergism, that is red, indicating there's really a push towards cytotoxicity. You can clearly visualize this all in one plot. And then we can also do single organoid analysis, like I previously mentioned, that this single organoid analysis can really capture the heterogeneity of a response within one organoid line and correlates very well to the progression-free survival of patients. As an output, we output a drug response table, so an Excel file that on top of the GR value also has relative viability, the GR metric, the NDR metric, and so on. And we can make any types of plots, custom reports, and so on. So if you're interested in implementing live cell imaging in your organoid drug screenings, I invite you to visit our website or contact us. And I'm also very happy to show you a sneak peek of the web application that we're currently developing. Here I have an overview of your experiments. And in this case, I'm going to show you the Orbits Oncology data that I used in the presentation. So for this experiment, we have 3 replicates of the same organoids. And you get a clear visualization of the different therapies on the plate map that you can upload very easily yourself. Here's a list of the different conditions, so the well name, the cell line, the treatments. Here you can have a quick look at your organoids if your screen is of good quality. And here you can again see the difference in morphology between hub organoids and OncoPro organoids. If you want to make sure that your image segmentation is adequate, you can also check this if it's sufficient. So the next step would be to upload the images. So for the last one, we did not upload images yet. We can easily do this from your computer. And this is a typical experiment in our lab. So we have a Steak and Spark Cyto, which outputs a folder with all the images for each time point separately. So we have a little tool, a desktop application that combines all the different time points, all the bright field fluorescence images and so on in a single H5 file for each well. So we can upload that. In the meantime, you can continue with the rest. So this one is currently analyzing. So here you can select the type of analysis you want, run it, and run it in the background. So once the analysis has been performed, we can also have a look at your downloads folder, which will have all the information that you will need. So you can have a data table. So we have now a pre-defined data table, but of course you can ask us to output any data that you want, giving all the time points, the replicates, the cell line, the conditions. It automatically calculates all the drug response metrics, relative viability, the NRGR metric, all the derivatives, the array over the curve, and so on. So everything you need, just one click on the analysis button, and you have all your data. So you can use this table to generate plots yourself, or you can use our predefined plots, as you can see here, to directly visualize the differences in response and look at your drug response. So this was a short overview of our web application that we have now. So we're very excited for this, really offering fully automated image and data analysis from image to data visualization and data tables. If you're not doing organoid drug screenings in-house, you can also make use of our new service platform at DrugVision.AI, where we will offer drug screenings on our patient-derived tumor organoid biobank. So feel free to contact us. So lastly, I want to thank all the people involved, and especially my core team, Maxime, Sophie, and Felicia, who really work really hard to make everything happen that I showed you in the presentation. Thank you very much for your attention.
All right. Thank you, Dr. Deben, for a really nice presentation with lots of very, very interesting work. And we will now begin the live Q&A portion of the webinar. So we've got lots of questions that have already come in. If you haven't submitted a question, please do so now. You can click on the Ask a Question box located on the far left of your screen, and we will answer as many questions as we have time for. So we can get started now. One question here, Christophe, for you. Can you elaborate a bit on the importance of distinguishing between cytotoxic and cytostatic effects in some of these screens, some of the advantages that the Orbits platform has to do that versus other methods?
Yeah, sure. So for us, it's really crucial that we can make the distinction because, especially for a lot of targeted therapies, we often see that, or TKIs, that they only induce cytostatic effect on the organoids, and often that's also seen in the clinic. So in our synergy studies, we're really looking for therapies that work in combination to push cells towards cell death, to hopefully get a better clinical response also. So I think it's really crucial for developing compounds that are really more effective in the clinic.
Thank you. And some of the question about some of the details of the analysis pipeline, particularly around how often imaging is required to feed into the system.
So now standardly we take image at 3 time points, so at least at time point 0, because we quantify the growth rate over time. And then at 72 hours and 120 hours. And most of the time we use the readouts at 120 hours. So I would say at least 2 time points.
We've got one question in about restrictions on commercializing models made using some of these reagents and licensing requirements. I can take that one. Thermo Fisher does not require a license for the commercial use of OncoPro medium, excluding any clinical, diagnostic, and further manufacturing applications. So we can't provide legal advice on the necessity of third-party licenses. I can tell you that the formulation has been specifically optimized without some components that are found in other systems, including Wnt, arspondin, and noggin, and any small molecule inhibitors. So we encourage you to consult with legal counsel for any legal guidance related to the product, but can emphasize that Thermo Fisher doesn't require license for use of the media, excluding those applications I mentioned. I will also say several of the lines that I discussed in my presentation are commercially available and can be used, and we know they're compatible with the system, and you can find out more about those at thermofisher.com/oncopro. We've got another question about breast cancer organoids. We have used OncoPro with breast cancer tumoroids. We'd have an application note that provides further details on that. We will typically supplement the base media with beta estradiol in the case of breast cancer, and I've seen that it works well with the bottles we've tested thus far. There's one more question about your data, Dr. Deben, showing some of the differences between media systems and how that may affect screening and results. Any comments from you on how that should be interpreted and kind of implications for what system should be used in different parts of the workflow?
I cannot give recommendations yet, but at least for us, it's now really important to still keep both culturing methods side by side and compare them and try to get more clinical data on it. So it might be that for some therapies, one medium works better than the other. So I think it's just an additional tool. Might not replace everything, but definitely in our insights, very valuable to have some different drug responses, because in the conventional method, we see that not every time there's a good translation into a clinical prediction.
Yeah, thank you. And we've certainly seen the same thing in our hands, even screening, performing some screens in OncoPro medium versus human plasma-like medium and some other basals. Seems like there's a good amount of flexibility in setting up the endpoint assays here. I think kind of some of the power of Dr. Deben's data set and throughput that you have is really being able to do so many screens and then match it up to some of the clinical data that's coming in. You also mentioned assembloids in your talk. Do you have experience using kind of co-culture type application or co-culture assays for these types of studies?
So recently we got a co-focal imaging system. So we're now really focusing on these co-culture experiments because we do see that using conventional wide field imaging, like we use for the tumor organoids, is not really sufficient to extract cell type specific responses. So there I would still advise to go forward with co-focal imaging.
Thank you. Yeah, from our side, we've done a little bit with co-culture with fibroblasts and endothelial cells. We've seen survival of those cells in OncoPro for a bit over a week thus far. Some more experiments are ongoing. It does seem compatible with some of those co-culture approaches. Certainly, cell tracking is a major concern when you get into those. There's a question about OncoPro medium containing Wnt. It does not contain any Wnt agonists built into the media system. All right, we might have time for one more question here. Someone says, I have cancer organoid lines from multiple donors that I want to screen in parallel. Do you have any advice for synchronizing cultures or otherwise ensuring consistent seeding between plates or donors?
Well, I don't think it's really necessary to screen them in parallel, but we do really use also live cell imaging and bright field imaging to perform counting of the organoids so that we have very accurate seeding density. So I think that's really crucial in your pipeline.
All right, very good. So thanks very much. I think we are about at time here. So I want to thank you again, Dr. Deben, for your time, for really interesting and
You're
important
welcome.
research. I would like to thank LabRoots as well as Thermo Fisher Scientific for underwriting the webinar today. And before we go, I'd like to invite everyone to answer a survey questions that you see on your screen to indicate if you're interested in receiving more information about OncoPro Medium or any of the tools mentioned today. So thank you to everyone who joined us for the great questions. Any questions that we didn't have time for or that were submitted during the on-demand period will be addressed by the speakers via the contact information you provided at the time of registration. This webinar can also be viewed on demand, and LabRoots will alert you by email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. So thank you again. Take care, everyone, and goodbye.
Bye.